In Chapter 8, we are going to continue our discussion of Mendelian genetics, except that we're going to apply the concepts that we learned to inheritance patterns that are not exactly following Mendel's rule. So in Chapter 8, we are going to talk about essentially unexpected phenotypes. So alleles and genes behave the same way no matter what. They are passed on in predictable manners, but sometimes we get phenotypes that we are not expecting. So we're going to talk about how we get unexpected phenotypes from a variety of gene interactions, environmental actions. We are also going to jump ahead just a little bit to Chapter 10 and do one section from Chapter 10 because I feel that it fits in here really nicely. All right, so as I said just a minute ago, genes, because they are located on chromosomes, behave in a very particular and predictable manner. Because chromosomes are passed on in a very specific fashion during meiosis, it follows that those alleles are passed on in a predictable way. And therefore, we can expect individuals to inherit specific genes and alleles from their parents. But sometimes, even though that inheritance pattern is predictable, we get phenotypes that we might not actually expect. And this is where we're going to deviate from Mendels Laws. Well, we're not going to deviate from Mendels Laws. Mendels Laws, the law of segregation and the law of independent assortment, are still going to apply here. But we're going to deviate from those phenotypic ratios that Gregor Mendel saw when he was looking at simple dominance. So there are several ways that we can get unexpected phenotypes and unexpected phenotypic ratios. First of all, we need to keep in mind that phenotypes arise from the interaction of alleles. So we saw this in the most simplest form when we were looking at Mendel's simple dominance. So essentially, if we have two alleles, let's say allele C, with allele C being the dominant one, we get predictable phenotypes. So we get the fully dominant phenotype, and then we get the recessive phenotype. Now, when we look at that heterozygote, we see that we have two different alleles, but one of the alleles is completely dominant. And therefore, it is covering up the other and controlling the phenotype of that individual. Sometimes that doesn't happen. Sometimes you get different interactions between alleles where the heterozygote has a different phenotype than you would expect. Basically, you get a third phenotype out of the heterozygote. You can also have actions of multiple genes. And again, we saw this in its most basic form when we were looking at dihybrids. Now, dihybrid crosses actually occur in this chapter, but I moved them into Chapter 5 because Chapter 5 is a little bit smaller. And with your basic dihybrid, you're still seeing very simple gene interactions. But when you do the dihybrid, you get combination phenotypes. So you get a variety of A and Bs together. But multiple genes can interact in different ways. And then on top of that, phenotypes can also be affected by the environment. And also something called penetrance, which is how strongly you see that phenotype, and expressivity, which is very similar. Essentially, why do some people or some organisms express it and others don't? So we're going to look at all these different types of interactions of alleles, interactions of multiple genes, and interactions from outside factors or from other factors. So we're going to talk about a lot of things in this chapter, but I wanted to give you a list of all of those things so that you have something to refer back to when you are studying, just sort of a checklist. So we've already talked about simple dominance. We did that in Chapter 5. That's also called complete dominance. So you might hear me say that or hear the book say complete dominance. This is when one allele fully covers up the other. We talked about dihybrid inheritance, essentially following the complete inheritance pattern. A little bit later, we're going to talk about linked genes. Now, these are genes that are located closely together on the same chromosome. This is compared to a dihybrid where those alleles and genes are located on different chromosomes, but we're going to come back to that later. In chapter 8, we'll talk about incomplete and codominance. We'll talk about ranked dominance of multiple alleles. We'll talk about pleiotropy, lethal alleles, and epistasis. Then we'll talk about conditional mutants, penetrance, expressivity, and then finally we'll end with complex traits and polygenic inheritance, which is our chapter 10 information, and the only information we're going to go over for chapter 10 in this semester. So before we move on, our book actually puts this information a little bit later, but I wanted to clarify a couple of things just so we're all on the same page. So of course we have genes. Genes are essentially controlling a particular characteristic. Within a particular gene, you might have different alleles. So alleles are different versions of a gene. So we saw these alleles when we talked in chapter 5 about the pea plants. This might be the yellow or the green seed, or the wrinkled or the round. The alleles are basically different versions of the type of gene you're looking at. So we will be talking about alleles quite often. Generally, those alleles, when we refer to alleles, we're talking about functional alleles. So these are basically different versions that are not mutations, because mutations are alleles that are typically non-functioning alleles. And usually they're rare. Very often they are also detrimental to the organism, although they don't have to be. But we will often refer to individuals with this non-functional allele as a mutant. So we'll sometimes say the normal or the wild type. We saw wild type when we did our fly lab, but we have normal and wild type, and then we have our mutant alleles, and generally we call those mutations. Polymorphism is very, very similar to alleles, except that it's a little bit more specific. So a polymorphism is more than one common allele, whereas when you're just talking about alleles, they may or may not all be equally common. A polymorphism tends to be a human vocabulary word, I guess. It's basically used to describe human traits generally. So a polymorphism is going to be something like blood types, where a lot of individuals have these alleles. They're more frequent in the population. And then something that we'll talk about, well, actually we won't talk about it in chapter 10, but I wanted to highlight the word because it's sort of applicable here, is the idea of a haplotype. Now, this is going to seem a little bit strange just thrown in here, but I wanted to talk about it because our book mentions it several times throughout, but we won't go over the chapter where we define it. So a haplotype is basically a group of genes and their alleles that are inherited together. So this is often used to describe human populations. So a group of individuals from a particular region might share a haplotype or a group of genes with specific alleles due to, say, a founding member in that region or a single parent in that region. So just sort of tuck that one away in your mind for a little bit. We're not going to talk about it in this chapter, but it does come up quite a bit, and it comes up particularly after chapter 10. And since we have one little section of chapter 10, I wanted to include that here. So let's dive right into these other unexpected phenotypes. So I find the books description of codominance a little bit confusing. So I'm going to go back to the way that I teach it in Biology 100 and Biology 101. And we are going to just look at phenotypes. Now, your book does have a chart that talks about incomplete dominance and codominance depending on what level you're looking at. And if you would like, I can speak with you individually on that chart. But we're going to go a little bit more basic here because there's so much going on in this chapter. So in codominance, this is where two alleles contribute equally to a phenotype. So in other words, you see both alleles. So both alleles are visible. So remember, in complete dominance or simple dominance, only one allele is visible in that heterozygote. So our example here is roan cattle. So roan refers to this phenotype where you have sort of this red and white together. So this fur color is controlled by two alleles at a particular locus, a red allele and a white allele. And in this codominant situation, they are both equally expressed and equally displayed. So instead of being red or white, we get sort of this mottled red and white. Just like Mendelian genetics, we can apply the use of a Punnett square. So let's say that this cow had parents who were red. So a homozygous red parent and a homozygous white parent. Now, if we were applying our idea of simple dominance, a heterozygote would have to be either red or white. Well, in codominance, we see that the heterozygote actually has a new phenotype. So in simple dominance, for two alleles, we would have two phenotypes. But in codominance, when we have two alleles, we actually get three phenotypes because that heterozygote has a new phenotype. So let's look at what's happening here. You might notice that I've used slightly different notation here. This is to indicate that there's a different type of inheritance going on. And I've used two capital letters to show you that these are essentially both dominant phenotypes. Sometimes in our book, I can't remember if our book writes it this way, sometimes codominance is actually written as a superscript. So you'll have a letter that indicates something like, say, fur color, and then the red is a superscript, or the W is a superscript to indicate that we're talking about fur color, but this is a new situation. So that you guys don't confuse codominance with the next example, I'm going to use this notation here. So let's cross these two cows this cow and a bull, a red one and a white one. And we'll see that all of the individuals are heterozygous. So all individuals are heterozygous genotype, and all are going to be roan, which is this mottled red and white. So let's say we cross a roan cattle or a roan cow with a white bull. So this roan is the heterozygote, so we're going to have RW cross the white, which is the WW. So let's see what happens here. So here we have two heterozygous individuals, so these are both going to be roan. And here we have two homozygous individuals, so these are going to be white. So in this cross, we have 2 heterozygous to 2 homozygous. We can reduce that to 1 to 1, and then of course that means we have 1 roan to 1 white, or half of our individuals are going to be roan and half of them white. Notice that there are no red individuals here, because these individuals that are heterozygote are not displaying fully red. In another very similar example, we can have something called incomplete dominance. So incomplete dominance is essentially the opposite of codominance. With incomplete dominance, neither allele can fully produce the phenotype, meaning that the heterozygote, again, has a different phenotype. So again, with Mendelian genetics, if we have 2 alleles, we expect 2 phenotypes. But here, we have 2 alleles, but 3 phenotypes again. So in incomplete dominance, basically you are producing, or you have one allele that is functional and the other one that is not. So in this case, we are going to look at snapdragons. These are a pretty little flower. They're kind of a cup-shaped flower that come in a lot of different colors. But in order to get the red pigment in the snapdragon, these red individuals are producing red pigment. So each of those alleles is producing a certain amount of red pigment. The white snapdragons produce no pigment. So this is a dysfunctional allele. It does not produce a protein that can give any pigment. So essentially they are lack of pigment flowers. In the heterozygote, when you only have one red allele, you still get red pigment, but you get less red pigment. So essentially this heterozygote is just a less red individual, but it appears pink because it's kind of a diluted pigment. So you get less red pigment, you get no contribution from that white allele. So let's say we have a red, well, we'll just practice with the previous one, a red cross white. So again, we're going to use some new notation here. We're going to use C for color, red. The red individual is the homozygous, cross C for color with the white allele. So when we write out this cross, this is really, really similar to that roan cattle example, except that our heterozygote gives a different phenotype. So all individuals are heterozygous, and all individuals are going to be pink. So let's do one more example. Let's cross a red, cross a pink, and see what we get. So red is our homozygous. So to do this, we could back cross. A back cross is where you take the F1 generation and cross it to the parental generation. So we could back cross, or we could use an individual from another line. So let's say we back cross to that parent. We'll write out our Punnett square here. So we get some homozygous red flowers and some heterozygous pink flowers. Now notice there are no white here. So we have one homozygous to one heterozygous, and we have 1-to-1 red to pink. So there we saw 2 alleles, and for everything we've done so far, we've been working with only 2 alleles. But you can actually have more than 2 alleles at any locus. Essentially, you could have as many as you want, 4, 5, 6, 7. We won't do anything quite that complicated, but I do want you to know that you can have multiple alleles. And with those multiple alleles, you often have what we call ranked dominance. So when you have those multiple alleles, you can get a variety of dominance. So you might have 4 of them, and you have one that is dominant over all of them, one that is recessive to the first but dominant to the other 2, and so on. So you might have allele A is dominant to allele A1, which is dominant to allele A2, which is dominant to allele A3, where A3 is the most recessive or the fully recessive, where you only see that if you have 2 copies of it. And then A being the most dominant, where you see that in the heterozygote no matter what your other alleles are. So to demonstrate this idea of multiple alleles, let's do a few examples. So we'll do an example using coat color in guinea pigs, which is controlled by 4 different alleles. So we've got the most dominant full color allele. So this is going to be a dark color, like a dark brown. We've got the sepia allele that produces a tan color. We've got a cream allele that is kind of a off-white color. And then we've got albino, which is going to be fully white. Now, the order of dominance is listed here, where full color is the most dominant, albino is the most recessive. So let's look at the sepia cross cream, where both had an albino parent. So that means that each one had a parent who was CA. So this sepia individual, let's look at that person. So they would have had a sepia parent. We don't know what the other allele was because it could have been any of the 3 lowercase c alleles, but we know that it had an albino parent. So it actually doesn't really matter what this parent's allele is, because the offspring is sepia, and if you have an albino parent, the only way you can get sepia is in a heterozygous. So there's our sepia individual. Let's do our cream individual now. So cream, this is going to be the same idea. Cream had a parent that was, we'll just assume cream, and a albino parent. So we know that they had one albino parent because this cream individual is one of the more recessive alleles. The other parent could have been somebody heterozygous. For simplicity's sake, we will just assume that they were either homozygous for cream or heterozygous for cream albino. It doesn't really matter because that individual passed on the cream from one, or received the allele for cream from one parent and the albino from another. So this is the cross we're doing here, CKCA cross CDCA. So let's write out that Punnett square, CKCA cross CDCA. We write or complete our Punnett square. I'm going to keep the more dominant allele on the left, just so we can keep track of what's going on here. So here we have 3 different alleles, and we're getting 3 different phenotypes. So half of our individuals are going to be sepia. So we're going to have 2 sepia. a quarter of our individuals are going to be cream. And then the remaining quarter are going to be albino. We have 3 phenotypes, but we have 4 genotypes. We have 1 CKCD to 1 CKCA to 1 CDCA to 1 homozygous for albino. So if you are not pausing to practice these on your own, I would suggest that you do that. Pause here, try the problem on your own, and then come back and watch me work it. So let's look at another sepia cross cream. I'm actually going to start with this cream individual here because I want to talk about the parents. So cream individual in our cross and comes from 2 sepia parents. So let's look at the parents. So they're going to be CK something cross CK something. We know they're sepia. Now, in order to make a cream individual, one of them has to pass on a cream allele. This other one could pass on a CD or a CA, because when this individual passes on that cream allele, they can be either CDCD or CDCA. Now let's look at that sepia from our cross. So that sepia had an albino parent. So we don't even need to write out what all of these crosses are, because this albino parent was CACA. So that means the sepia individual is going to be CKCA, because this albino parent had to give the A, and obviously we get a sepia from the other parent. So what cross are we going to do here? Well, basically there are 2 different ways you can, or 2 different potentials. Unless you know what this cream individual actually is, you probably need to do both crosses to see what the potential offspring are. So in one cross, we have CD cross the CKCA from the other parent. So we've got a CKCD, we've got 2 of those, so these are going to be sepia. And then we've got a CDCA and a CDCD. So these are both going to be cream. Now the other potential cross is if the other parent passed on an albino allele. So our other potential cross is CDCA cross CKCA. So let's see what we get there. We get CKCD, CKCA, so these guys are going to be sepia, CDCA. That individual is going to be cream, and this one is going to be albino. So unless we're told what this parent was, we are unsure what the genotype is for that cream. Now, I understand that this can get a little complicated. On an exam, I will most likely be a little bit more clear about what allele you're going to use, or I might try to be a little tricky and see if you can catch this. So be ready for both. Make sure that you're thinking about what alleles you could possibly get. So I'm going to skip example 3 real quick and go over to example 4. 3 is the most complicated, and so we're going to do 4 first. All right, so again, sepia crossed cream. So let's start with the sepia parent. So I'm sorry, the sepia individual. So sepia said sepia had a full color parent and an albino. So full color, that's a capital C there, cross an albino parent. So in order to get this individual to be sepia, this allele has to be the K allele. So this sepia individual is going to be CKCA. So that's our sepia. Now let's look at the cream. Now we're not told anything about the cream parents. Oh yes we are. Hold on, let's go back. If we look at number 4, oh nope. Hold on. When I made my PowerPoint, I missed copying a little bit. So cream, we can add this in here, and cream had 2 full color parents. All right, so if the cream individual had 2 full color parents, we know we have big C for both of the parents. Now to get cream, one of them must give a cream allele. This one could give the cream or, excuse me, the albino, I did it again, the albino allele. Let's just pick one. This is going to be exactly like problem 2, where you have to you could potentially do both of the crosses. For time's sake and simplicity's sake, I'm going to say that this one is heterozygous for the albino. So this individual, the offspring, if they are cream, they must be CDCA. Now, the other option would be that they must be CDCD if you chose this version, and you certainly could do both of those. I'm just going to do one for time's sake. So let's do cross our sepia heterozygous with our cream heterozygous. So we've got a sepia, a cream, and a cream. I'm sorry, an albino. So we have 2 sepia to 1 cream to 1 albino. Now, for practice, I would like you to do the cross where this was the parent. So we would have CKCA cross CDCD and see what you get. So let's go back to problem 3. I saved this one for last because it is the most complicated of them. So again, we're going to cross our sepia and cream. Let's start with our sepia individual. So this sepia had 2 full color parents. So that means that each of those parents has a full color allele. Now, because they are full color, these parents each could be CCCD, CCK, or CCA. Now, we don't know what they are, so that makes determining what this sepia individual is very challenging. We know that one of them has to have that sepia allele, but the other one, we're not sure if they are, or we're not sure which of these they are. So this sepia individual could be CDCD, could be CDCK, or could be CDCA. So there's a lot of possibilities here. Now let's go to our cream. So the cream had 2 sepia parents. So we know that one of the parents has to be CK, CD. And the other one is CD, we're not sure. Could either be CD, CD, or CD, CA. Now, the challenge here is determining which crosses we need to do. So we're actually going to use a little bit of logic here, and rather than actually write out all of the crosses, we're going to sort of FOIL these and see what potential offspring we could get. So if our, actually I'm getting a little bit ahead of myself. Let's figure out what the cream genotypes actually are. I'm sorry, your cream, yes. I'm confusing myself. See, these are why this is confusing. Our cream individuals could be CD, CD, or CD, CA. So let's go. If our individual is CD, CD, cross this guy, we get cream individuals, and they're all cream. Now, if we cross this individual, if that individual has the homozygous, we get either sepia or cream again, right? And then if this individual, or if we are homozygous and we cross it here, we are going to get cream again or albino. So I'm just sort of looking at these and determining what potential genotypes we could have. Let's look at the other potential phenotype. So if we are this one, the heterozygote, and we cross with that one, our options are cream or, actually, it's only cream. Cream's the only option because this guy has only the cream alleles, and that's dominant over the albino. If we make this cross, we can again get cream or we can get sepia. So again, we can get cream or sepia. Now this one, if we do this one here, we can get cream or we can get albino. So depending on what alleles those grandparents pass down, you can get any combination of cream, sepia, and albino. Now, if you would like to figure out the actual ratios, you're welcome to figure this all out. It is quite complicated. Or if you want to just for practice do one of these, say maybe you want to do the CDCK cross CDCA. You can practice any of these just to get some more practice with the multiple alleles if you would like. I'm not going to do all of those crosses. We'll just say in summary that you can get any of these four. The only thing you can't get is no full color because you don't have any full color alleles. Now, of course, what offspring you actually do get depends on the genotype of those sepia and cream, and they can be one of many.